Thank you very much, Dan. Can you hear me?
Yep.
Yael Chanin, Karen Abraham, Artium, Inval, I see Ronit Sadik by now. Well, thanks so much for arranging this beautiful summer school. When I spoke to Dan about giving the talk, he made it difficult, and he asked me not to talk about research we're doing in the lab, and mainly to talk about the general field. And as we all know, scientists love talking about the research they do, and that's painful. One thing is not talking about your own research. It becomes much more painful when you need to talk about research other people are doing, and I'll do my best. So thanks so much for coming in. In the next 45 minutes, I'd like to discuss how can we actually improve our drug delivery approaches, or maybe some lessons we've learned in the past by choosing a drug delivery system. So we'll start with drugs, and then we'll continue to siRNA. So drugs, I mean small molecules, and siRNA, nucleic acids of the different forms. Some lessons other people have learned, some of the lessons we have learned, and how they can actually translate and maybe help us here advance the field. So when we talk about small molecule delivery, Doxil comes to mind. Doxil, I'm sure, was mentioned here already several times. I heard it in the last talk. It's a 100 nanometer liposome with doxorubicin remotely loaded into these particles. So this is a doxorubicin crystal inside of the 100 nanometer liposome, and it's used today for treating nearly 300,000 breast and ovarian cancer patients, as you know. So the building blocks for Doxil are actually three main building blocks that became very, very important or highly used in the field of drug delivery initially. And today, I think people are trying to find new tools and new vehicles. So HSPC is the main building block. The two hydrophobic tails, the Schroder ionic head, the high phase transition temperature around 52 degrees Celsius is actually what will give us the structure of the sphere. And then two components that are actually critical for preventing the drug from leaking out of the liposome. So we have cholesterol acting as cement between the building blocks and PEG DSPE. So these are the PEG heads, and the DSPE anchor that anchors into the lipid bilayer. And anyone here in drug delivery, I'm sure, is well aware of this system. When you come to formulate or to synthesize liposomes, there's several approaches. One of them, which is becoming also highly used, is the ethanol injection approach. You dissolve the lipids in ethanol, you hydrate them to form multilamellar vesicles or small unilamellar vesicles, and you actually, if you want to use an extrusion system, you move on from these micron scale onion-like particles to 100 nanometer liposomes. You can also configure them slightly larger or slightly smaller. So the extruder, again, a system that was in use for many years and seems to be phasing out. Some of the new technologies will be discussed here later on in this conference. But for an extrusion system, you have your sample. It's highly pressurized and then forced through membranes to downsize the liposomes to the target size. And unlike regular filtration membranes that look like a mesh, when we talk about extrusion, we use polycarbonate membranes that are actually etched. You can notice the holes in the membrane that actually configure the particles to have a very uniform size. So going back to our lipid vesicle, if it's a liposome, it's a sphere, something like a tennis ball with hair coming out of it. And you can notice these are the PEG entities actually increase circulation time. The half-life in the blood without PEG can be less than 2 hours, and with PEG can reach up to 72 hours. We can load hydrophilic compounds into the aqueous core, hydrophobic compounds to the membrane, and proteins across the membrane. We can add targeting moieties to the surface of these systems. And since Doxil was approved, it was approved for several different indications. It became a very attractive system for trying to deliver drugs. So that's what many groups tried doing. They actually took one liposome that was already FDA approved and tried to load different drugs into these liposomes. One major example is cisplatin, another anti-cancer drug. So doxorubicin actually worked very well in the clinic. And this steroid reached the clinic, but has not yet been fully translated. And Chezi Bellenholtz, who may be speaking later on today, may discuss this system in more detail. But if we go down to cisplatin, you can notice the molecule. It's a rather gentle compound. And if it's dissolved, for example, in DMSO, it loses its biological activity. And it has very, very low solubility in water. So several approaches have been used to actually load large amounts of cisplatin into liposomes, primarily changing the solubility limit of the compound by raising the temperature of the solution. And cisplatin actually changes its solubility limit approximately threefold between 20 degrees C and 70 or 80 degrees Celsius. But this approach of trying to use one vehicle for many different drugs didn't really translate. So we're talking about cisplatin. When I put up the slide, it actually, I like the X here because it really didn't work well. This is what the liposomes would look like. This is doxorubicin. You can notice the large amount of drug actually per particle. Again, an osmotic pump is used to load these particles after they are formed. So first you form an empty liposome, then you pump in the drug, and large amounts are inside. Unlike liposomal cisplatin, in which approximately 1/10 of the amount of drug can actually be loaded into each of these vesicles. And phase 1 and 2 trials, several of them actually failed. And the conclusion, you can read right down here, SPI 77, this was the liposomal cisplatin, is essentially inactive. And that's the main bottom line. And it's inactive because the drug just doesn't release from the drug delivery system. So it targets, there's no question about it. You see high accumulation in the tumor sites. However, none of the drug is actually available because it sits so comfortably inside of the liposomes. And that's where other groups actually kicked in, trying to alter the initial liposomal formulation to a formulation that would release the drug in a more therapeutic form. So one approach is to alter the membrane, and other groups actually looked at different systems, and now I'm talking about delivery of different compounds. And the PEG entity we have on the surface of the liposomes. So again, these PEG hairs shield the particle through hydration. They actually disguise, and these liposomes are called, known as stealth. They disguise them in circulation, extending half-life. However, sometimes we do want the immune system to recognize our liposome formulation rather rapidly. And if we formulate the particles without the PEG, we'll see agglomeration in the vial before they're injected to the patients or to the animal. So how can we actually play with both considerations? One, have a stable formulation, and two, also remove these PEG entities as they're injected. So Kinnick, I think one of the pioneers in the field of siRNA delivery, chose a very simple approach. Instead of using a PEG DSP system that has a very long anchor into the lipid bilayer, they used PEG chains that have actually a very short anchor into the lipid bilayer, C14, 14 carbons, and immediately after injecting it into circulation, the PEG sheds off and the particle is actually exposed. So now instead of having the PEG shield the particle, you have a particle that's unshielded, and it has totally different biodistribution in the body. Also, some reaches the liver site, some reaches the brain. And in this case, it's an ApoE targeting mechanism. So that would be altering the PEG. And we know that altering the membrane surely will change also the efficacy of drug delivery systems. So if we shift out for 1 second from lipid-based particles, we have here PLGA nanoparticle was actually approved by the FDA, but discontinued in 2004. You can notice this beautiful Nature Biotechnology paper that came out on this, regarding the system. And when they were asked why they actually discontinued, why the company pulled back the system, which seemed to have some therapeutic efficacy, you can notice here, it's actually very painful when you inject these nanoparticles to the patient. So these reactions included nodules. Nodules is in a small lump after the injection. 61% of the patients. Erythema, 53%, and they're on. So sometimes you can develop this beautiful drug delivery system, but if it's painful for the patient when it's introduced, that may actually cause the company to come and pull back. And these are considerations we have to keep in mind. So we have to remember there's a patient at the end of the line, and the technology has to actually be able to be compatible with that patient, so the patient would actually come back and really want to be treated again. Another type of nanoparticle that's out there, if we were already talking about polymer-based systems, we can't not mention the albumin-based Abraxane with sales that exceed, I think, $1 billion or $2 billion a year. It seems to be one of the most powerful nanotechnologies that's out there. These particles are, again, around 100, 110 nanometers, and they have an active targeting mechanism when they reach the cell. However, it still remains very unknown how these systems interact with cancer. Surely they also follow the EPR, which was mentioned earlier today. However, once they reach the target sites, they see there is some uptake that seems to be active uptake into the particle. So one, we see that one size won't fit all, and we have to tailor our systems for the drug itself. But when we look at gene delivery, several other considerations also come to mind. So just to lay the ground, when we discuss gene delivery, we have I would say one major approach, two components. We have some cationic system that physically actually binds the negatively charged nucleic acid. It can be DNA or RNA, and this is where most of the science is actually taking place. Very few uncharged particles for delivering DNA or RNA out there, but studies have been performed, and I think the main reason people like these systems is that you get a lot more loading of the charged DNA or RNA in your particle when you use a physical system. So we're discussing DNA or RNA delivery, and we can't discuss such a system without mentioning the first patient that actually passed away due to gene delivery. Jesse Gelsinger, an 18-year-old student, couldn't eat steak or have beer. And that was his disease. It's called OTCD, and he was offered to have a gene therapy approach for trying to correct his problem. It was supposed to be targeted to the liver, and an adenoviral vector was actually used for carrying the gene to his liver. So this is what the father actually described. This is after Jesse passed away. I received a call from the principal investigator on Monday, just after they infused Jesse. The father was, I think he was not in the hospital at the time. He explained that everything went well and that Jesse would return to his room in a few hours. I discussed the infusion and how the vector did its job. He explained that if they could affect about 1% of Jesse's cells, they would get the results they desired. So this was his father. And I think, putting aside the very sad outcome of this gene therapy treatment, there is an opportunity here. And if this is correct, that we don't need to really treat every cell and cell in the diseased tissue, we can only treat a certain percentage of the cell, I think it really increases our chances of bringing gene therapy to the clinic and bringing nanotechnologies that will actually carry genes to the patient. So many times we need to begin our thoughts with considerations about the disease. Would gene therapy even be effective there? we want to select the proper therapeutic compound, and then we also have this opportunity that we may not have to treat every cell in the body to reach efficacy. And this is the second story that actually began rather sadly but ended quietly but very, very happily. So the SCID examples of gene delivery, or the first ones are well known. These are immunodeficient children. And again, here there's a gene delivery approach using either different types of vectors, and we'll discuss the vectors in 1 second. And this, I think, again, in the audience, everyone is well aware of. This is a scientific paper that came out. These children, many of them developed leukemia, and these are actually the news reports that harmed the entire field of drug delivery after these failed trials with SCID children. However, when we look back at the path of research, the two very important, I think, points that maybe some of the researchers put aside when we were developing such systems. And so a U.S. study last year looked at the long-term effect of infecting the IL-2 gene into mice. A third of the animals developed a form of cancer, with most were about 10 months old. So this may be some clue to what may happen later on to these patients right here. And the second study that somehow was overcome by the regulatory bodies that were supposed to approve such a clinical trial, a few years previously, a French trial was halted prematurely after 4 of 11 boys treated were diagnosed with T-cell leukemia. So I think many times we see things that happen in the lab when we deliver our systems, when we develop drug delivery systems. And we have to remember, if at the end of the day we really want these technologies to be translated and reach patients, we have to look at these clues and ask ourselves, is it really safe what we're developing out there, and what would be the outcomes if they're not safe? So in this case, I think some of the safety data was overlooked. Also looking at the construct, what gene we're taking there, and also at the carrier. So those are two aspects. And some of us may be experts in the construct, but not in the carrier. And some are vice versa, experts in the carrier and not in the construct. And that's where we need to work together. And I think one of the nice things the field of nanotechnology really highlights is the diversity of researchers working together to try and reach an active result. So this paper was published 2 years ago in Science Translational Medicine, and it actually highlights all the successes that have been made since those failed trials early on. So these trials were mainly done rather quietly, and so gene therapy, SCID, X1, great success. Some of the kids actually have survival of 9 years and more since then, but they were looking back at the lessons we had in the past to try and deliver better technologies. So when we try to develop new vehicles for delivering genes, we actually have some competition. Retroviruses, adenoviruses, and adeno-associated viruses, which grant certain advantages. So retroviruses are hardly used today. Adeno-associated viruses are widely used. They're small particles. You can notice both of these are small particles, less than 100 nanometers. And they have a rather mild immuno stimulation. So these issues have been overcome. They don't integrate into the host genome. Many concerns that we had from gene delivery systems are actually were actually overcome, and they're approved. So China was the first actually to approve them. There it's used for delivering P53, and I think we'll be seeing more and more of these gene delivery systems in there. So where is there still room for us when we come to invent? And I think it's in the size of the construct. So you can only deliver very small constructs using these systems, so less than 8 kilobase pairs. And many times the genes we actually want to deliver are larger. Plus, we have to tackle this issue of immunostimulation, especially if we're looking at repetitive injections. And here, Don, I think, is really one of the world experts in this field. So a whole bunch of different cationic systems have been developed for delivering genes. And a list of guidelines have actually been published to try and define what we're looking, where we need to protect our construct, and thereon, and it ends with the safety and maybe also biodegradability of the systems we are developing. And I think if we take one step back and we look at the immunostimulation, Don and his group here, Dan Per, published this paper in Biomaterials that actually highlights the mechanism by which cationic systems can instigate the immune systems, and why some of them would actually probably never reach a therapeutic reality. So what can you do actually to modulate some of the some of the immuno activity of these systems? A paper that came out last year actually pointed it out, and we used this approach a couple years earlier, is actually using less charge on our binding agent. So instead of using a highly cationic system for strong complexation with the DNA, we actually use a system that has charges that are spread out and not as dense. And what we find is actually the DNA doesn't bind that strongly to the particle, which plays a double role: one in the immunostimulation, and two in the efficacy of these systems. So I'm a chemical engineer by training, and I had to choose a system that would be compatible with a chemical engineer. That means chemistry that isn't too sophisticated. And I was looking at this polyethylene amine molecule, and it's you can notice the cationic charges granted by the amine. However, PEI is rather toxic if it's long, and if it's short, it was shown to be ineffective for gene delivery. So this is the chemical engineering chemistry, and it's epoxide ring opening conjugation. This ring opens, and you get a lipid tail that actually conjugates onto a polymer backbone. So we have many different types of lipids we can conjugate to the polymer backbone, and we selected to work with actually a short one. However, in general, we could look at PEIs that are of different lengths and have different amounts of lipid tails, and also lipid tails of different lengths. So we chose a PEI that was 500 in molecular weight and lipid tails of different orders. We screened many different compounds, and we found those that were best for carrying siRNA into HeLa cells and actually knocking down selectively one gene. Then we moved on because we know HeLa cells are really considered to be rather simple for transfecting. We added 2 additional cell lines, and we found again the best hits, those compounds that are good also in HeLa and also in additional cell lines. And we took it on to an in vivo study, which we recently published, and again to find those compounds that were most effective in carrying siRNA into the body and knocking down a gene with a high efficacy, but also at very low doses. So we're talking at 0.02 milligrams per kilogram of the RNA. What do these particles look like? So these aren't liposomes for sure. This is a totally different system. The particles are around 60 to 70 nanometers in diameter, and you can notice they have a structure that resembles a multilamellar liposome. However, it's all in the nanoscale. So it's an onion-like structure with the RNA actually loaded between each of these layers. So we asked ourselves, why would some particles be more effective in delivering RNA while others would be less effective? All the particles were actually taken up by the cells, and we saw no difference in that aspect, so it had to be something else. And what we found is actually the strength of binding between the lipid bilayer of the particle, or between the bilayer of the particle and the RNA actually dictates if this system will be efficacious or non-efficacious. If we're able to deliver the RNA but it's not released inside of the cell, then of course we won't get any therapeutic activity, just like with the cisplatin that was injected to patients. However, we have to have these two contradictory requirements from our system. One is that we're able to protect the RNA before reaching the target site, and two, once we're at the target site, we have to be able to release it in an effective manner. And maybe just in a poetic view, you know, we want to hold on, but not hold on too tight. If we hold on too tight, it probably won't work well. And this is, I think, the next generation of delivery, just highlighting the need to deliver effectively into cells and into organelles inside of cells. This is a picture from Ronit Sachi-Feinaro's group here. I think one of the nicest images I've seen come out. And actually looking at, can we target different organelles inside of cells and maybe affect therapeutic efficacy using that approach. Today we know that there may be another aspect that plays in delivery of RNA. It's not only the uptake of the particles into the cell, it's also the excretion of the particles before they actually are active. So we'll see uptake, but then we'll see some of the particles are expelled from the cell, and that means they'll never perform any therapeutic activity. So we need to enter the cell, lodge at our target site, and release the therapeutic compound on site. So this is a workshop, and I would want to say a couple of words about the importance of lipid formulation. Chezi Barnot just entered, and he led this study. We were looking at liposomes from a different perspective. In this case, we were trying to identify if liposomes could act not as drug delivery vehicles, but could act as biomaterials for reducing the friction inside of joints. And this was done together with Dorit Nitzan and Yitzhak Etzion from the Technion. So we're looking at the liposomes now not as a system for delivering a drug, but a system that may be able to be functional inside the body. And the disease we were trying to tackle is osteoarthritis. You can notice here a healthy joint, and on the right-hand side you can notice an osteoarthritic joint. It actually lacks the cartilage that lubricates between the two bones. This is also very painful. Many people, I'm sure you know, suffer from this disorder. And we were thinking, could you actually inject particles that would sit here in this interface between the two bones and reduce friction and reduce pain later on? So when you look back at the science of joints, it actually has phospholipids inside of it. So a healthy joint will have phospholipids that reduce friction. We have also other components such as hyaluronic acid, which is mainly degraded under inflammatory conditions, proteins, carbohydrates, and the phospholipids have been shown to play a major role in protecting cartilage health. If we look at them, these are the same phospholipids, are similar to those that we actually use for formulating liposomes. However, they're lacking when we look at patients that have osteoarthritis. So could we refurbish the joint by supplementing with exogenous lipids? The system that was constructed by Yitzhak Etzion was a cartilage-on-cartilage system, and which was able to actually measure different friction coefficients between cartilage using the different liposomes under different media, different loads that would resemble different joints or different weights of the patient, and different motions right and left that would resemble walking or running or climbing upstairs. The cartilage we used for these studies was sourced from human patients, and actually this is what I think helped bring the system later on to the clinic, and Chezi may want to comment later today about this system. So we screened a great deal of phospholipids, and you can notice the major difference is actually in the tail length or in the saturation of these lipids. So tail length of 14 carbons without any double bonds would grant you a phase transition temperature of 23 degrees. And we mentioned HSPC earlier in the lecture, which has a phase transition temperature of 52, meaning it's a harder lipid at body temperature, while this lipid would actually be rather soft at body temperature. And schematically, this is what it looks like. This would be a hard lipid bilayer, and this would be a soft one. And we asked ourselves, would any of these formulations actually affect cartilage lubrication? And surprisingly, we found that if the lipids are actually soft, it's better. We have lower friction coefficients, while if the lipid bilayer is hard, we get higher friction inside of the joints. And extensive studies we conducted actually found that not having a hard layer, and not a soft one, but actually having a combination of soft and hard granted us the best properties we wanted from the drug delivery system. And this is actually granted by mixing two different lipids. One would be soft at the body temperature, and one would be hard at the body temperature. In this case, unlike previously, microparticles were far more effective than nano. The microparticles sat on the surface of the cartilage, while the nanoparticles penetrated deeply inside. And this is what was able to actually lubricate. If the particles would be deep in cartilage, nothing would actually protect the surface during the therapeutic cycle. So we have a system where you have cartilage that's protected with lipid bilayers that are actually semi-soft and semi-hard. They can absorb the energy, but they're firm enough to actually maintain themselves under the body motion. So this was translated to the clinic. And you can notice the comparison between hyaluronic acid injections and liposomal injections. Hyaluronic acid is the gold standard today in osteoarthritis and the liposomal system. And what we were actually, not us, what the company, Moebius Medical, was actually measuring is the quality of life of these patients after they were injected. Could they climb stairs? Could they get up and down? How much pain were they suffering? And you can notice the improvement in their activity over time after receiving the liposomes in a local injection into the joint. So what we assume is that the lipid formulation here plays a major role, actually, and only having the right balance inside the lipids would actually grant you the low friction and also the minimization of pain in cartilage motion. And this was actually also noticed by the amount of pain medication the patients actually consumed. So you can notice patients that receive these liposomes in a local injection consume less pain medication over time in comparison to those that received the hyaluronic acid injection. And as an engineer, I think it's wonderful. You see the science come together and really improve the lives of patients, and hopefully we'll be able to improve the lives of many more later on. So I want to just end, because I promised not to talk about our work too much, and I'd want to just end with two approaches we have today for treating cancer. This will be like 4 or 5 slides, that's it. We discussed delivering DNA and delivering RNA. However, there's a lot of interest today in delivering protein. And if we're looking, it's not only delivering protein, we were actually looking at delivering molecular machines that can perform a therapeutic task on site. So you could actually deliver a system that will be able to do something once it reaches the target site. And the dogma we had in our mind was particles that would be injected into circulation. They would reach a target site and then perform a therapeutic task. And I'll mention just 2 therapeutic tasks that we were looking at. One was to synthesize a drug on site, so particles that would have all the molecular machines inside of them and could be triggered from outside the body to produce a drug wherever it's necessary. And 2, a system we just showed a proof of concept in vivo in which a particle reaches the target site and performs a therapeutic task of actually cutting tissue. So a local surgery with molecular precision at the disease site. So in this case, we'd have a drug that would actually be produced on site. This is one of these particles that are actually made of DMPC, so it's a very soft lipid composition. And the reason it's soft is when we try to work with harder lipid compositions, we many times damage some of the molecular machines we try to load into these vesicles. And the second aspect we have to have is the control aspect. So in this case, we use DNA. This is added by us. The machinery also added by us. However, the DNA dictates which protein would be synthesized on site. So we'd have a DNA strand. It has a switch which turns it on or off. In this case, it's a cage, and we're working on an additional cage or additional system. You'd actually, once you hit this particle with light, the DNA is freed. It's inserted into the machinery, and protein is produced. In this case, a therapeutic protein. And you can notice here the production of a protein inside the body in animals that were exposed to light in comparison to animals that were not exposed to that trigger. So the second aspect is actually cutting tissue on site. So particles actually perform a therapeutic task inside the body. And what we looked at again is the evolution of surgical tools. You can notice this stone, 4,000 years old. Also in the Bible, we have mentioning of stones used for cutting tissue. Our scalpel today isn't very different, but biology has actually evolved far more advanced than the systems we have today for cutting tissue, because biology or enzymes are actually capable of detecting a specific molecule. So we have molecular precision for cutting tissue, and we just completed our first proof of concept here inside of rats, being capable of cutting tissue with molecular precision using a system that actually can work on site. I'd like to conclude with several thoughts. It's a workshop, so we're not trying to highlight, I think, our work or other people's work. We're trying to bring together many strong minds and think how can we advance the entire field. And sometimes, what delivery system properties grant the greatest benefit to justify clinical translation? Are we really all interested in clinical translation? I think not all of us, and that may not be our goal, and that's okay. Just like the previous speaker that mentioned the carbon nanotubes, they may not reach the clinic. I'm not sure. I hold slightly different opinions, but they're surely great tools for probing at the cellular level and understanding. That's where we can also grant a lot of information. And I think we're all looking towards an era of personalized medicine. So can we do personalized targeting? That would be one question. And two, can we use our systems really to try and differentiate between one patient and another, try and improve therapy in that approach? And I'll end here with great thanks to you and to the students of my team. Thank you.